Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. From today onwards, I am going to start a series of lectures on enzymes. We will see the clinical significance of enzymes and all the details about the enzymes. So for today's lecture, this is first part of this series of lectures. So today we will see the basics of enzymes. We will see the classification of enzymes. Again, basics of classification, and then we'll see the active sites of the enzyme. In this series of lectures, we are going to cover first two parts, that is introduction and nomenclature. In the course of this series, we will talk about enzymes and characteristic features, factors affecting the enzymes in details. Then we will see enzyme inhibition, all the different types of inhibitions, where we will see different type of reactions like different type of equations. We will see enzymes regulations also. We will talk about the enzymes specificity, cooperativity. We will talk about the clinical significance of enzymes. There we will talk about the diagnostic enzymes also. And lastly, we will talk about the isoenzymes. So this is a long series of lectures. We will have 8 or 9 different lectures. So let's start today's section. Enzymes are normally called as biocatalyst, or it's also called as catalyst of life. But why it's if you see this biocatalyst, bio means life and catalyst means any substance that speed up the chemical reaction without being a reactant is called as catalyst. So the catalyst is defined as the substance that increases the velocity or rate of reactions without itself undergoing any changes. Means at the end of the reactions, the enzymes will be unchanged, or catalyst should be unchanged. So that is called as catalyst. Any catalyst which catalyzes the reaction in the biological system in the living cells, they are called as biocatalysts, and that biocatalysts are called as enzymes. So enzymes are biocatalysts. Enzymes may be defined as a biocatalyst synthesized by the living cells. I have already told you. Almost all the enzymes, I'm telling almost all, not exclusively, almost all the enzymes are protein in nature. There are certain exceptions like means RNA acting as an enzyme are called as ribozymes. So any RNA which works as an enzyme are called as ribozymes. One of the example is spliceosome, which normally perform during the RNA processing, during the transcription, means when RNA is going to be synthesized. So RNA processing, their spliceosome functions. All these enzymes are colloidal in nature. They are thermolevel in characteristic feature. Thermolevel means they are temperature sensitive and very very specific in their actions. So enzymes are, they are having a stereo specificity also, which we will talk later on. In the laboratory, hydrolysis of protein by a strong acid at 100 degrees C takes place at least a couple of days. But if you will perform same reactions at in the presence of enzyme which occurs in our GIT tract, that takes only few hours. So if you will perform any reaction without any enzymes, it takes days. But when enzymes will be there, the reaction will be going to finish within hours. So that is the function, that is the beauty of enzymes. When these enzymes perform a critical task of lowering the reaction, means how any catalyst or any enzymes perform their functions. So if we'll see, normally enzymes perform the critical task of lowering the reactions activation energy. Now what is activation energy? Activation is the amount of energy which normally requires for any reactions to perform. If you will say, this is the reactant energy and this is the product energy. Normally whenever we are having a normal chemical reactions, reactant converted to product, and this reaction is a spontaneous reaction because energy of reactant is higher and energy of product is lower. So for this reaction to perform, we need a activation energy of this one. This is the line of, this is the base of the reactant energy. So before starting the reactions, energy should go high, and this is the minimum threshold value which a substrate needs to start the reaction, and then it will come back to this one. So normally this is the rate activation energy. What exactly enzyme is doing? Enzyme is going to decrease the activation energy requirement. So energy requirement for the reactions, previously this much amount of energy was requiring, but in the presence of enzyme, only this much amount of energy is needed. So ultimately enzyme is going to reduce the activation energy requirement. So amount that must be put in the reaction to begin. Enzymes works by putting the reactant molecule and holding them in such a way so that the chemical bond breaking and the formation bond breaking and the bond formation process takes place very efficiently, very easily manner. Another point you need to remember is enzymes does not change the reactions G delta G value. So if you see, this is the delta G. So in the presence of energy also, in the absence of energy enzyme, sorry, in the presence of enzyme also, in the absence of enzyme also, this delta G is not going to have any kind of changes. So they don't change whether the reaction is energy releasing or energy absorbing or any conditions. It is not going to change at all. Enzymes lowers the energy of the transition state. And what is transition state? Transition state is an unstable state that products must pass through in order to become a reactant. So this is how enzymes functions. Now, if we'll go towards the historical background of the enzymes, the first thing is Berzelius. In 1836, he coined the term catalysis. So the term catalysis was coined by Berzelius in 1836. Then later on in 1878, Kuhne was another scientist who used the term enzyme. In Greek, the enzyme means which is called as yeast. So Kuhne used the term enzymes to indicate the catalysis which is taking place in the biological system. Then isolation of enzyme system from a self extract from the yeast was achieved in 1883 by Buchner, and he coined the term zymage. Later on, this was the found that this is the mixture of enzymes. This is not a single enzyme. Later on, it was found that zymage is the mixture of enzymes which convert sugar into alcohol. And then James Sumner came in 1926, who first achieved the isolation and crystallization of the enzymes urease from the jack bean and identified them as a protein. So this is roughly a historical scientific. In medical, many competitive examinations, this some of the questions comes from this portion, like who had first crystallized the enzyme, who has first isolated the enzymes, who coined the term different. So this time, if you'll see the nomenclature of the enzymes, normally in the early days, they were given names by their discoverers. And in an arbitrary manner, we don't have any scientific nomenclature system. Like pepsin, trypsin, chymotrypsin, these does not have any system. Anything, any, you can call it any procedures to nomenclature, do put name like that one. Sometimes we are going to add suffix age. If we see in the previous cases, like age, like catalase. So these are the we are going to use suffix age in the name of the substrate. So whatever name of the substrate will be there, you will add age. It means it's called as their enzymes which digest them, like lactase for lactose, lipase for lipids, nuclease for nucleic acids. So these are called as trivial names. But again, we didn't have a system like uh, either it is on rough category or on the basis of substrate. But if you'll see what is the scientific nomenclature systems, that name has come from by IUB system of enzyme classification. There we are having a scientific nomenclature system that we are going to see it. Other than that one, enzyme is also classified into broad category, which is called as intracellular and extracellular enzymes. Intracellular enzymes means all those enzymes which normally functions within the periphery, inside the periphery of plasma membrane, where they are synthesized. So any enzymes which function inside the cells, inside the plasma membrane, are called as intracellular, and any other enzymes which is normally functions outside the cells, outside the plasma membrane, are called as extracellular enzymes. Roughly almost all the digestive enzymes belongs to these extracellular groups of enzymes. However, both intracellular and extracellular enzymes are synthesized inside the cells, inside the plasma membrane only. Then the International Union of Biochemistry appointed an enzyme commission in 1961. In 1964, this International Union of Biochemistry system of enzyme classification had come into the force for their classification and nomenclature. So enzymes are divided into the six major classes by those IUB system of enzyme classification. But currently we are having a seventh class also. So at present we are having total seven classes of enzyme. Each class of have their own its own represent and the general type of reaction brought about by the enzymes in that classes. So what are those classes? If you'll say the first class of enzyme is called as oxidoreductase. Normally, this type of enzymes which is involved in the oxidation reduction reactions, they catalyze the redox reaction and can be categorized into oxidase or reductase. Like here, one substrate is going to be reduced, another substrate is going to be oxidized. So there will be reaction where oxidation reduction is going to going to be associated. Some of the enzymes in this category are alcohol dehydrogenase, or any D-amino oxidase, or cytochrome oxidase, or reductase. Second class of enzyme is called as transferase. This normally transfer of a functional group. So this catalyzes the transfer or exchange of certain groups among some of the substances. Like if you see here, this is one substrate and this is another substrate. When two substrate reacts, some portion of the substrate from one substrate is going to be transferred into the other substrate. So here B is going to be transferred from A to C. So this is called as transferase. Some of the example of this transferase category is hexokinase. Almost all the kinases which transfer phosphate from ATP or another one component to other component, like transmethylase, phosphorylase, transaminases. So these are called like transferase enzyme which transfer certain functional group from one place to other place. Third category is hydrolase, which normally hydrolysis of the various compound. So this accelerates the hydrolysis of a substance where in the presence of water, substrate is going to be broken down into two or more than two different fragments. Enzymes, some of the enzymes which belongs to hydrolase category are lipase, choline esterase, or alkaline phosphatase, pepsin, urease. Then the fourth class is lyase. So these enzymes specialize in the addition or removal of water, ammonia, or carbon dioxide. So it promotes the removal of a group from a substrate to leave the double bond reaction and catalyzes its reverse reaction. So if you'll see here, this is a reversible reaction and it catalyzes something need to be removed. Either it may be oxygen, water, it may be carbon dioxide, it may be water molecules or ammonia or anything else. Some of the example of this lyase are aldolase, or it may be fumarase or histidase. The fifth class is called as isomerase, which normally involves in the reaction of isomerization reactions. Isomerization means only interchanges of the molecule or group or atoms. So it facilitates the conversion of isomers, geometric isomers or optical isomers. Like here you can see, previously this compound was arranged in the manner of A, B, C. So everything is same, only their arrangement has been changed. Like it becomes ACB. Some of the enzymes of this class is triose phosphate isomerase, or retinol isomerase, or phosphohexose isomerase. Sixth class is called as ligase. Ligase enzymes catalyze the synthetic reactions where two or more molecules are joined together in the presence of ATP. So it catalyzes the synthesis of two molecule substance into the one molecular compound. Some of the ligases are glutamine synthase, succinate thiokinase, or acetyl coenzyme carboxylase. And the seventh class is translocase, which normally helps. This is the newly added classes, which catalyzes the movement of ions or the any molecules across the plasma membrane or separation within the membrane. So this is the basic classification, 7 classes of enzymes. So in this sequential manner only these enzymes are classified. This is the first class, this is the second class. So this is the sequence is very important. Why I'm going to tell sequence is important? Because when we will talk about the IUB system of enzyme classification, this is called as class 1, this is class 2. So if we'll say these are the 7 classes, how you can remember these enzyme classes? So for we are having a mnemonics that is called as OR Halil T, oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase, and translocases. If we'll see the enzymes nomenclature in this case, we will have a separate video on the detailed enzyme classification schemes and their nomenclature. So every enzyme is having a IUPAC nomenclature system where every enzyme will have 4-digit val numbers. So if we will talk about oxidoreductase, so this will have a 4-digit number, that is called as IUPAC nomenclature. So we will talk in, or you can call it enzyme commission number. So later on we will have a separate video that there we will talk about the detailed classification scheme of this one. Now if you'll see, the chemical nature of enzymes are, all the enzymes are invariably proteins in nature except ribozyme, which is RNA molecule works as enzyme, and that is chemically spliceogen, which normally works during the RNA splicing processes. Each enzyme has their own tertiary structure and a specific conformation, which is very essential for its catalytic activity. Why tertiary structure is important? Because protein, you know, the protein is having 4 different structures on basis of their arrangement. It's primary, secondary, tertiary, and quaternary. Almost all the proteins are functional in their tertiary or quaternary structures. So enzymes active site forms, the active site becomes active only in their tertiary structure or quaternary structure. So this any enzymes, the functional unit of enzymes or any enzyme which is called as functional enzymes are known as holoenzymes. So holoenzymes are the functional unit of enzymes, which is composed made up of 2 different parts. It may be, there are 2 broad category of enzymes where one holoenzyme means only proteinous part is going to make it enzymes. So these holoenzymes have 2 different portions. One is called as apoenzymes, that is the proteinous part, and second portion is called as coenzymes or cofactor, which is a non-proteinous organic part. When these two portion combine together, apoenzyme and coenzymes, it is going to give us a holoenzyme. So holoenzyme is the functional enzymes, but this is not necessary for every cases. In some cases we don't require any cofactors. In that case, apoenzyme or means only proteinous part alone is sufficient to catalyze the reaction. So if in that case apoenzyme is equal to holoenzyme. But most of the cases, every enzyme needs certain kind of factors, certain kind of vitamins or certain non-proteinous parts portions, that is called as coenzyme or cofactor. So that is apoenzyme plus coenzyme equal to holoenzyme in that cases. Then if we'll say, the term prosthetic group we are going to use when we are talking about the enzymes activity. So the prosthetic group is used when non-proteinous moiety tightly associated with the apoenzymes. So if you'll see this portions, this apoenzyme associated with the coenzyme or coenzyme associated with the apoenzymes in 2 different fashions. Either it will be very tightly or very loosely associated. If this is very tightly associated with the covalent bond, so the non-proteinous part is called as prosthetic group. The coenzyme can be separated by dialysis from the enzymes, while the prosthetic group cannot be, because prosthetic groups are very tightly covalently associated with the apoenzymes. Then we are also using some terms, like one of the terms is called as monomeric enzymes, and this is used when if it is made up of a single polypeptide molecules, that is called as only one peptide will be there. That is called as like some of the examples are ribonuclease or trypsin. But some proteins have a multiple unit or more than one unit polypeptide chains. They are called as oligomeric enzymes. The examples are lactate dehydrogenase, aspartate transcarbamylase. In these enzymes, we can have, or the functional unit will have more than one polypeptide chains. So they are oligomeric enzymes. There are certain multi-enzyme complex also we are having, which catalyze a different reaction in the sequence. So only the native intact multi-enzyme complex is functionally active and not the individual units. Like if you'll say, one of the enzyme is pyruvate dehydrogenase, PDH, which is normally connecting the glycolysis along with the Krebs cycle. In that case, enzyme is having 3 important proteins, or three. So single protein, if will three out of these 3 polypeptides, even a single polypeptide will be there, that is non-functional. So till those all 3 proteins will come together, they cannot function it. So pyruvate dehydrogenase, fatty acid synthase, prostaglandin synthase, these are called as multi-enzyme complex. Means when all the polypeptide will be together, then only it will be functional. So we are having certain specific terms like holoenzymes, apoenzymes, cofactors, prosthetic groups, monomeric enzymes, oligomeric enzymes, multi-enzyme complex. So these things you need to remember. Then there are a number of factors are there which regulates, which affects the enzyme's activity. So these factors are enzyme concentration, substrate concentration, product concentration, temperature, pH or hydrogen ion concentration, presence or absence of activator or inhibitors, presence or repressor or de-repressors, covalent modifications. So all these factors we are going to see in details one by one along with their uh, graphs and the pictures what we are having in the separate video. So we will see all the factors which are going to affect the enzyme activity. Some of the examples just to review few things is like if you see the temperature. When we are going to see the temperature as a factor in the enzyme regulations, in that case, velocity of enzyme reaction increases with the increase in temperature. Like if you see, as you are going to increase the temperature, rate of reaction increases. At a certain point, the pH means enzyme activity will be the highest, and then if you increase the temperature, its activity is going to reduce. So this we are going to get a bell shape curve. Bell shape curve. So and this point the max peak point is called as optimum temperature. Similarly, in the case of pH also we are having a bell shape curve. If you will increase slowly pH, activity will increase. At a certain point it will be the optimum highest activity, and then it will decrease. So there also we are going to get a bell shape curve. The enzyme concentration, if you will see, as the concentration of enzyme is increases, the normally the velocity of reaction increases. uh, so normally we are going to get a straight curve, STD, in case of substance level. But in this case, substrate is also going to have a function. So that we will talk later. In the same time, when we are talking about the substrate concentrations, rate of reaction increases along as we are going to increase the substrate concentration. But at a certain point of time, even substrate concentration does not affect rate of reaction. So there will be a STD or plateau in the graph. So these all these factors we will see in the details later on. Now comes to the active site. What is this active site? Uh, Enzymes are big in size compared to the substrate, which are relatively smaller. So enzymes size, overall molecular weight and size is very high compared to the substrate. A small portion of huge enzyme molecules is directly involved in the substrate binding and catalysis. So the active site of an enzyme represents the small region at which the substrate binds and participates in the catalysis. So those portions on the enzyme molecules where substrate binds and a reaction takes place, that is called as active site. And this active site function is going to be functional only when protein or enzymes will be in the tertiary structure of the proteins. This active site is made up of a number of amino acids, which is called as catalytic residue or catalytic amino acids. These amino acids may be placed at one moment, one means all together, or it may be distantly located. One like if you, for example, if you say in case of lysozymes, which is having a 129 amino acids. So their active sites are going to be formed by 5 different amino acids, and these amino acids location will be at the 35th position, 52nd position, 62 position, 63 position, and 101 position. So if you compare this amino 63 and 101, they are very 40 amino acid, roughly 40 amino acid distantly placed. So the catalytic residue of an active site of an enzyme can be placed together or can be very distantly placed. These active sites are not rigid structure, they are very flexible. Where we will talk about the enzymes basic mechanism, there we will talk about the different kind of rules and different kind of theories. So there we will say enzymes active sites are flexible in nature. Generally, these active sites possess a substrate binding site and a catalytic site. Means there are two sites you can call it. One place where substrate binds, and it might be the same place or it might be a different place where actual reaction occurs. So, place where substrate binds are called as substrate binding site, and the place where actual reaction takes place are called as catalytic site. These substrate binding site and catalytic site can be same or it can be different. The commonly found amino acids at the active sites are serine, aspartate, cysteine, lysine, arginine, glutamine, or tyrosine. But if you'll see in this case, serine is the most commonly or most prominently found amino acids in the active sites. The active sites are regarded as a clefts and the crevices, or you can also call it pocket uh, on the big enzyme sites. The sometimes the enzymes means when enzymes requires cofactor or the coenzymes uh, are part of the catalytic site, not the substrate binding site. If enzymes have substrate binding site and catalytic site are different, so the cofactors is going to be placed at the catalytic site, not at the substrate binding site. Whenever substrate binds to this active site, the binding is normally very through the very weak non-covalent bonds. And enzymes are very specific in their function due to the existence of the active site only. So these are some of the important points regarding to the active sites. If we'll say, in this case, this is the substrate, this is the active site. Substrate binds to the active site. So substrate entering active site of the enzymes, it binds. There will be very weak interactions, weak bonding will be there, and as soon as it binds, there is a enzyme product formation will be there, and products is going to be living, and enzymes will be unchanged. It will be as such previously will be there. In this case, because substrate binds here, this is called a substrate binding place. The reactions will occur because in this case reaction occurs at the same point. So this is called catalytic site also. So the substrate binding sites and catalytic sites are the same in this case, but it may be different in different cases. So binding site where substrate binds and orient substrates, and the catalytic site where normally involved in the reduction of the chemical activation energy. So these are the two important places in the active sites. Later on in the other video, in the further video, we will talk about the detailed classification of the enzymes, and then we will talk about the factors affecting the enzyme's activity. So that is coming into the next video. So thanks you for the watching, and please like the video if you like. If you're really interested in the video, you can like the video. You can comments, you can write the suggestions if you need any kind of improvement, any other things, any specific video, you can write in the comment box. You can share with your friends, your colleagues, and if you're not subscribed my channel, you can subscribe my channels. Thank you very much. Have a nice day.